Now that we've learned about the major players in the synapse and some of the terminology that goes along with them, we can begin to discuss, well, what happens when the action potential reaches the terminal bouton of the presynaptic cell. There's a chain of events that occur in this, and the whole reason we spent time talking about the action potential prior to this was so that we could understand how it leads to the release of neurotransmitter from the presynaptic cell. So that's what we will discuss presently. Release of transmitter from the presynaptic cell. First thing we're going to do is have the action potential arrive at the terminal bouton. And what is this? What do we mean, action potential? We talk about it almost like it's an object. But of course it's not an object. It's a flow of current. It's a flow of positive charge. And that positive charge is going to make its way all the way down to the terminal bouton. And in the terminal bouton, it's going to cause calcium channels to open up. And of course calcium will flow through those channels. The influx of this calcium will cause the synaptic vesicle to fuse with the presynaptic membrane. And this is actually a curious issue in neuroscience right now. No one's actually sure why calcium is necessary. What is calcium doing to help the vesicles bind to the presynaptic membrane? And there are some theories on it, but nobody's demonstrated convincingly one way or the other what it is that calcium's doing. All we know is that when you plug up these calcium channels, the neurotransmitter won't release. The vesicles will not bind to the cell membrane. So nobody knows exactly what calcium's doing, but we do know that it needs to be there. Now, through the process of exocytosis, which is also brought on by the influx of this calcium, the vesicle is going to be turned inside out, dumping its contents into the synaptic cleft. So again, we have a vesicle full of neurotransmitter. It's floating around in the synaptic terminal. When calcium comes in to the presynaptic terminal, thanks to the arrival of the action potential, the vesicles will bind or fuse to the cell membrane, and then they will turn themselves inside out so that the neurotransmitter that was inside of them goes suddenly from being inside the terminal bouton to outside into the synaptic cleft. And we then take this synaptic vesicle back into the presynaptic terminal bouton to package more neurotransmitter. So this is in effect what is happening in the presynaptic cell upon the arrival of the action potential. I'll point you to two videos that are posted on Vista in Module 2, Synaptic Transmission, and there are actually two videos on synaptic transmission. One will talk about enzymatic deactivation, and the other video will talk about reuptake. So I'll just briefly here foreshadow what those videos will tell you. Enzymatic deactivation and reuptake are two methods for getting neurotransmitter out of the cleft. You might ask, well, we just talked about how we get neurotransmitter into the cleft. Why are we talking about how to get it out now? Well, the issue is that the neurotransmitter is going to float across that synaptic cleft from the presynaptic cell over to the postsynaptic cell, and it's going to, in some cases, excite that postsynaptic neuron. It's going to cause it to get really excited and start firing action potentials. But there is a such thing as firing too much. In fact, a neuron can die. It's called excitotoxicity, toxicity from excitation. The cell will actually start to produce, waste that will kill it. So we have to have some mechanism for getting the neurotransmitter out of the cleft after it's done its work. And it turns out we have two mechanisms for that. Enzymatic deactivation just refers to the introduction into the cleft of an enzyme. And this enzyme has the power to split the neurotransmitter into its constituent components. For instance, acetylcholine, I believe, is the neurotransmitter that's discussed in this enzymatic deactivation video. Acetylcholine is made up of two molecules, acetate and choline. You put the two together and you have acetylcholine. But you take the two of them apart and it's not acetylcholine, it's acetate and it's choline. And so acetate and choline on its own will not excite the postsynaptic cell. Acetylcholine, though, if you bind the two together, that compound can excite the postsynaptic cell. So, once you've allowed your acetylcholine to excite the postsynaptic cell for long enough, then you're going to send your enzymes into the cleft to parse or cleave these compounds into their constituent components. And now all you have is acetate and choline floating around in there. They won't bind to the receptors in the postsynaptic cell, and they therefore will not excite the postsynaptic cell. So that's one way of getting the enzyme out of getting the neurotransmitter out of the cleft. A second way is to use the process of reuptake. And this may sound familiar to you if you happen to know anything about certain antidepressants like SSRIs, selective serotonin reuptake inhibitors. Selective serotonin reuptake inhibitors. So it should be obvious right now that what an SSRI is doing is inhibiting the reuptake of serotonin into the presynaptic cell. So that is the second method by which we can remove excess neurotransmitters from the cleft, is by transporting them back across the membrane back into the presynaptic cell. And so in the case of depression and anxiety, it's often the case that there is not enough serotonin in the person's system. So what can you do to instead of trying to increase the amount of serotonin in the person's brain, you could just slow down these reuptake transporters. And if they're slowed down, then the serotonin that's in the cleft is going to stay there for a longer period of time, and therefore give you more of the effect on the postsynaptic cells that you need from that serotonin. So again, selective serotonin reuptake inhibitors are selectively inhibiting the reuptake of serotonin. And this helps us remember that reuptake is one of the two processes by which we can take excess neurotransmitter out of the synaptic cleft. Having discussed the process of exocytosis and how the neurotransmitter diffuses from the presynaptic cell across the cleft to the postsynaptic cell, let's now discuss what effect that neurotransmitter has on the postsynaptic cell. And what's going to happen is these neurotransmitters are going to create postsynaptic potentials. That is, we're going to have changes in electrical charge potential in the postsynaptic cell. And we have two basic kinds. The first kind is an excitatory postsynaptic potential. And the most common way to achieve an excitatory postsynaptic potential is by having a neurotransmitter that opens sodium channels in the postsynaptic cell. Now, note that these sodium channels are not voltage gated. They're not voltage gated. They're not voltage dependent. They are transmitter gated or transmitter dependent. That is, it's not a change in the voltage of the cell membrane that causes these sodium channels to open. Rather, it's a binding of a neurotransmitter to receptors on those sodium channels that causes them to open. And again, as we already know, if we open a sodium channel, sodium is going to follow both its concentration gradient and its voltage gradient from the outside of the cell to the inside of the cell. And if we're bringing positive charge into the cell, that means we're going more positive. And the other type of potential is an inhibitory postsynaptic potential. And this usually involves either chloride entering the cell through transmitter-gated chlorine channels, or it involves potassium leaving the cell through transmitter-gated potassium channels. Now, let's look at the EPSP and the IPSP separately in more So in the case of the excitatory postsynaptic potential, we're going to have neurotransmitter bind to receptors on the transmitter-gated sodium channels in the postsynaptic cell. And that is going to allow sodium to flow into the cell. When the channel opens, sodium rushes in to the postsynaptic cell. And if enough sodium enters the cell will depolarize to the excitation threshold of -55 millivolts. So we see that over here on the right in this graph. We see that the cell is going along at its resting potential of -65 millivolts, and allowing sodium to flow into the cell causes depolarization. What you do not see in the graph here is enough depolarization to actually get us to the excitation threshold. If we do get to the excitation threshold, then this function you see in the graph here is not going to go back down immediately to resting baseline. Rather, it's going to go all the way up to +40 millivolts and then back down to form an actual action potential. So if we reach the excitation threshold, that is, we get enough positively charged sodium flowing into the neuron, then the postsynaptic cell will fire an action potential in its axon. And what's going on is the positive charge that has entered the dendrites is going to propagate all the way down to the axon. And once that positive charge reaches the sodium channels, if it's enough positive charge, it causes those sodium channels to open, kicking off that saltatory conduction of the action potential that we discussed previously. Now the inhibitory postsynaptic potential, forgive me, I notice up here at top I have excitatory. I will change that to inhibitory. If in the case of an inhibitory postsynaptic potential, we can call that an IPSP. Neurotransmitter binds to the receptors on transmitter-gated chloride channels, in this case. The channels open and allow chloride to rush into the postsynaptic cell. And if enough chloride enters, cells will depolarize to hyperpolarize to -75 microvolts. And in this case, instead of moving toward excitation threshold, we're actually moving away from excitation threshold. And we're going to cause the cell to not fire an action potential because we're moving further away from the excitation threshold as opposed to moving closer to it. So we now understand how the neurotransmitter binds to receptors in the postsynaptic cell, causing either sodium to rush in, chloride to rush in, or potassium to rush out, and how these lead to either excitatory postsynaptic potentials and inhibitory postsynaptic potentials. But the fact is, any single postsynaptic potential is not going to be enough to cause any significant change in the postsynaptic cell. That is, just because we have one cell firing one action potential, releasing one volley of neurotransmitter onto the postsynaptic cell, that may create, say, an excitatory postsynaptic potential, but it's going to be a very minimal one on its own. We really need that neuron, either that neuron to fire many action potentials in a very short amount of time, releasing a lot of neurotransmitter, creating a lot of excitatory postsynaptic potentials, or we need multiple neurons at different locations on that cell to release to fire multiple action potentials, releasing multiple volleys of neurotransmitter, creating multiple excitatory postsynaptic potentials. And if one of those two things happens, or both, then what we get is something called summation. We have those two kinds of summation. The first I discussed just now was temporal summation. And this is the idea that a single neuron, for example in this image below, neuron number 2, if it fires just a single action potential and releases neurotransmitter onto the bronze-colored postsynaptic cell here, the target neuron as it's described in this image, then that single release of neurotransmitter will create an excitatory postsynaptic potential in the target neuron, but it won't be enough to reach excitation threshold. But if we can get that neuron number 2 to fire multiple action potentials in a short amount of time, thereby releasing more neurotransmitter, thereby creating more excitatory postsynaptic potentials in the target neuron, then that should be enough positive charge to reach the axon and cause the sodium channels to depolarize to their excitation threshold, thereby causing the target neuron to fire an action potential. So that's temporal summation, multiple PSPs at one location in a short period of time. The other kind of summation is spatial summation. And this is where we have multiple postsynaptic potentials being created simultaneously at multiple locations in the cell. So in the case of this figure we have here, if we imagine that neurons 1 and 2, which are synapsing at different spatial locations on the postsynaptic cell, on the target neuron, if they fire action potentials simultaneously at different locations, then they will cause excitatory postsynaptic potentials, multiple ones, to occur at different locations in the neuron, but those two postsynaptic potentials will move into the cell body and they will add together, they will sum together. And if there's enough summation, that postsynaptic potential will reach the sodium channels in the axon, causing them to depolarize to their excitation threshold, enabling them to open up and initiate an action potential in the postsynaptic neuron, in the axon of the postsynaptic neuron. Now, what I've described here is a process that's gone over in a fair amount of detail in a video that's posted on Vista. In fact, this image that we have on this page is an image, a screenshot of that video. So it's very difficult for me to really describe this in much detail in this kind of static imagery I have here in this PowerPoint. So I'm going to have to rely on you to make sure you go to Vista and watch this video several times. Very important to get it down. And in fact, if you look over here on the right side of the image, you'll see these blue-colored inhibitory neurons. And what we can see that in that case, what these neurons are doing, the blue neurons are releasing a neurotransmitter that actually is going to cause the opening of either chloride channels, which allows negative charge to rush into the cell, or we open potassium channels, which are going to cause positive charge to rush out of the cell. Either way, you've got inhibition. That is, if you've got a lot of negative charge coming into the cell gets more negative, it hyperpolarizes. Or if you have a lot of positive charge in the form of potassium flowing out of the cell, then that's also going to make the inside of the cell more negative, creating an inhibitory postsynaptic potential. So let's review what we've done here. And I'm going to base this review on the videos that you have. We have the resting potential, the action potential, saltatory conduction, synaptic transmission, and the summation of excitatory and inhibitory postsynaptic potentials. So the first video we watched in this series was the one about the resting membrane potential, and this explained the relative concentrations of sodium and chloride outside of the cell versus potassium inside the cell. It talked about the concentration gradient and the voltage gradients, and how in the case of sodium, the concentration and voltage gradients work in unison with one another, whereas with chloride and with potassium, the concentration and voltage gradients push in opposite directions. We then looked at the video discussing how action potentials get started, and we saw that if we had enough positive charge reach these yellow-colored sodium channels, those sodium channels, in other words, reached excitation threshold, those sodium channels opened up, allowing positive charge to rush into the cell, so much positive charge that we go from a negative 55 millivolt resting or excitation threshold up to a positive 40 millivolt peak in the action potential. Then we go back down our falling phase of the action potential to where we get to negative 65 millivolts, and then even go below it, we hyperpolarize and eventually return back to the resting level of negative 65 millivolts. And then we looked at the video discussing how the myelination created by oligodendrocytes on the axons helps us both keep the wires from getting crossed, that is, axons of different neurons from mixing their signals up with one another, and more importantly, how the myelinated segments enable the light speed conduction of the electrical current, and how we have nodes of Ranvier in between each one of these myelin sheaths in order to regenerate the axon, or excuse me, in order to regenerate the action potential. And so this video compared the propagation of an action potential down an unmyelinated axon at the top of the video screen compared to the propagation of an action potential through a myelinated axon at the bottom of the video screen. And then the next thing we discussed was synaptic transmission. We noted how the action potential arriving at the axon terminal in the terminal bouton will cause the initiation of exocytosis, whereby the neurotransmitter in synaptic vesicles are turned inside out and flushed into the synaptic cleft, how the neurotransmitter will go across the synaptic cleft to bind to receptors in the postsynaptic cell. And then we discussed how we can get that neurotransmitter out of the synaptic cleft either by enzymatic deactivation or through reuptake, and then how new neurotransmitter vesicles are filled with their neurotransmitters. And one thing I forgot to mention here is that calcium plays a role in this. That is, the action potential runs down into the terminal bouton, causing voltage-gated calcium channels to open up. And again, nobody knows exactly why this calcium is important, but we do know that it is necessary to get this process of exocytosis. And then finally, we just discussed the video on inhibitory and excitatory postsynaptic potentials and how those sum together.